Hello students, this is Dr. Sujata Magdum, Assistant Professor in Zoology, KTHM College, Nashik. I'll be dealing with biological chemistry for third year BSc student, and the topic of discussion is colloid classification and their uses, which is from chapter 1, that is Introduction to Biological Chemistry. If you recall, in the previous lectures, we have talked about various types of bonds which are involved in structuring the macromolecules, as well as we have talked about the colloids in general. So today we shall move on further with only classification aspect of colloids and their uses. Colloids, as you all know, are the substances which are seen dispersed in other substances. These dispersed substances can be classified as the dispersion phase or the discontinuous phase, which are always seen mingled with the dispersion medium, which forms the continuous medium. Now, this colloid can be classified on various different aspects, and the classification is as follows. Like, the first one is based on the physical states of dispersed phase and the dispersion medium. Then the second aspect of classification is based on the interaction between the dispersed phase and the dispersion medium. The third aspect is on the basis of the size of the molecule or particles or the atoms of dispersed phase. And the fourth one is on the basis of its appearance, whereas lastly, it is classified on the basis of electric charges which are seen present on the dispersion phase. Now let us move ahead and understand each of these classification in detail. To begin with, the first aspect is based on physical state of dispersion phase and dispersion medium, wherein both the conditions, both the components of the colloid, that is the dispersion phase as well as dispersion medium, is considered. Here, the state of dispersion phase, it could be solid, it could be liquid, or it could be gases, and in the same way, the dispersion medium could also be solid, liquid, and gases. Depending on their interaction, or depending on their combination, I should say, we can classify them as solid sol, aerosol, gel, emulsion, liquid aerosol, solid foam, foam, or none. Now, to understand them, let us go into the detail. Dispersion phase, if it is solid, and if the dispersion medium is also solid, then the colloid what we get is called as a solid sol. The best example is alloys or the cranberry glasses, right? Now the next one, if the dispersion phase is solid, whereas the dispersion medium is liquid, then what we get is the sol, and best example is ink, and biological example you can quote is the blood. In the same manner, if the dispersion phase is solid, dispersion phase is solid, dispersion medium is gas, then what we get as a colloid is called as a aerosol, and the example is smoke or ice cloud. In the same manner, if the dispersion phase becomes liquid and the dispersion medium is solid, then the name given to such colloid is called as a gel. Example is jelly, and the biological or example is the curd. Similarly, if the dispersion phase is liquid and the dispersion medium also is liquid, then we get emulsion. Example is milk and cream. If the dispersion phase is liquid, whereas the dispersion medium is gas, we get liquid aerosol, and the example is cloud or fog. In the same manner, if the dispersion phase becomes gas and the dispersion medium is solid, what we get is the solid foam. Example is aerogel or pumic stone. In the same manner, if the dispersion phase is gas and the dispersion medium is liquid, we get foam. Example is shaving cream. And lastly, if both the dispersion phase as well as the dispersion medium are in the gaseous state, there we call them as the colloid and the best example is the gases. Now the second part or the second type of classification, which is based on observing the nature of interaction between the dispersed phase and the dispersion phase, we can classify colloids into two types, that is lipophilic and lyophobic. Now lyophilic is those wherein the dispersion phase has high affinity with that of the dispersion medium. In the same way, if we talk about lyophobic, phobic refers to hatred. So hence here we can say that the dispersion phase has low affinity with that of the dispersion medium. Further, because in case of lyophilic phase, there is high affinity to dispersion medium, we can say that the dispersion phase has high tendency to pass in colloidal solution, whereas lyophilic solutions, they don't have tendency to pass. Further, lyophilic are much more stable and lyophobic are relatively unstable. Further to state, the lyophilic sols or lyophilic colloids, these are reversible in nature and are self-stabilized, whereas lyophobic, they are irreversible in nature and stabilization is always done by addition of certain electrolyte. Lastly, if the dispersion medium in case of lyophilic solutions is water, then such lyophilic colloids are called as hydrophilic or emulsoids. In the same manner, if the dispersion medium in case of lyophobic sols is water, then it is called as hydrophobic or it is called as suspension. Best example to describe lyophilic is the starch, jelly, rubber and proteins, whereas lyophilic examples are soles of metals, soles of metal hydroxides or soles of metal sulfides. Next, the third way of classification of colloids is based on by observing the size of the molecule or the particle or the atom of dispersion phase. Depending on that, we can classify them as multimolecular colloids or macromolecular colloids. Multimolecular colloids are those colloids where the individual particle or the atom size of dispersion phase is less than 10 raised to minus 7 centimeters in diameter. Whereas in case of macromolecular colloids, the individual particle or molecule or atom size of dispersion phase is always greater than 10 raised to minus 7 centimeters in diameter. You can also remember macro refers to large, whereas multimolecular will contain less diameter of the colloidal phase, dispersion phase. For in multimolecular colloids, the particles are always held by van der Waals forces, and same holds true with the macromolecular colloids also. Example to describe multimolecular colloid is gold sol, whereas macromolecular colloids are the natural polymers, which includes large number of fibers like the silk fibers, cotton and so on. Now, the fourth aspect of classifying colloid is based on the appearance. So here we can say that the colloids can be classified as the sols or the gels. Sols are those which contains the fluid appearance or which shows the fluid appearance, whereas gels are the colloidal solution which appears to be solid in nature. Nomenclature is based on the dispersion medium. Here once again, in case of appearance of dispersion phase of colloids. Example is hydrosol or aquasol, wherein we can say the dispersion medium is water. If the dispersion medium is alcohol, then it is called as alcohol. If the dispersion medium happens to be benzene, then it is called as benzosol. Whereas examples of gels are jelly, butter, cheese and curd. Lastly, the classification of colloid is based on the electric charge on the dispersion phase. Now the electric charges could be either positive or negative, and based on this we can classify these colloids as the positive colloids and the negative colloids. In case of positive colloid, the individual particles or the molecules or the atoms of dispersion phase, they always carry a positive charge. Whereas in case of negative colloid, the individual particles or the molecules, or you can call them as atoms of dispersion phase, they always carry a negative charge. Example of positive colloid is metal hydroxide, and the biological example for positive colloids are hemoglobin or the basic dyes which we commonly use in the lab, that is methylene blue. And in nature, you know that proteins, they are always seen in soluble condition, or uh, I mean to say the soluble proteins, not about the structural proteins. So those soluble proteins which are seen in acidic media, they form the positive colloids. In the same manner, the best examples to describe negative colloids are the silver or copper soles and the base acidic dyes which we use in the lab, example like the eosin and the Congo red. And in the same manner, the proteins which are seen in soluble condition, and if the medium is basic, it gives rise to negative colloids. With this we come across, or we have completed the classification part of the colloidal solutions. Now we shall come to the next aspects of colloid and will understand the uses of colloid. The first one is it is used in medicine. Medicine in colloid form are easily absorbed by the body tissue and hence are more effective. Then the second is sewage disposal. Colloidal particles of the dirt, mud, etc. carry electric charges, and hence when sewage water is passed through the plates kept at very high potential, the colloidal particles are coagulated due to electrophoresis and the suspended matter gets removed. And this way the process of sewage disposal becomes very easy. These are also used in purification of water. The precipitation of colloidal impurities present in water can be done by adding certain electrolyte like alum, which is commonly used at our home, and the negatively charged colloidal particles of impurities get neutralized by the aluminum ions and settle down, and pure water can be decanted off. Further, these colloids are also used in cleansing action of soap. Soap solutions are actually the colloidal solutions. It removes the dirt particles either by adsorption or by emulsifying the greasy matter which is seen stuck with the clothes. So this way the process of cleansing can be observed by the action of soap. Then the formation of delta. You know delta, these are always seen associated with the river basins which are merging with the sea. Now river water contains charged colloidal particles of clay, sand and many other materials. Sea water is very high storage of variety of electrolytes dissolved in it. As soon as this river water it comes in contact with the sea water, then the water gets coagulated and coagulated in the sense the substances which are present both in water, pure water, that is river water, gets coagulated and gets suspended and ultimately settle down at the point of contact. This results in the rise in the river bed, and as a result water adopts a different course and delta is formed, and in due course of time there is mingling of water of both river as well as sea observed. Smoke precipitation. Now smoke precipitation we can understand by um, discussing with the aspect that is smoke particles. Now, smoke particles, these are actually electrically charged particles of carbon in air. Precipitation of smoke particle is carried out by the precipitator, which is based on the principle of electrophoresis. Smoke is allowed to pass through the chamber having a number of metal plates attached to a metal wire connected to a very high source of potential. All the charged particles of the smoke will get attracted by oppositely charged electrode and will get precipitated, and after losing their charge and the hot air passes out through the chimney. The dust particles are also removed in this process. Thus, the nuisance of the smoke which is created in the big industries can be avoided. Then this colloid is also used as a solution in photography. That is, the silver bromide solution in gelatin is applied on glass plates or celluloid films or papers to form the sensitive photoplates, and hence we can say it is a very important component in photography also. Also, these colloidal solutions, they are useful in uh, recovery of, or to come to what you can say, uh, to get relief from the deficient rains, or in other way we can say it is useful in creating artificial rains. Artificial rain can be caused by spraying oppositely charged colloidal dust or sand particles over a cloud. The colloidal water particle present in the cloud will be neutralized and coagulate to form bigger water droplets, and thus it causes rain. Then these colloids, they are also used in rubber industries. Example, latex, which is a colloidal solution of negatively charged rubber particle. From latex, rubber can be obtained by coagulation. Rubber plates or any article which is made from the rubber can be prepared by depositing negatively charged rubber particle over the article, and hence it is used to make the make the articles of rubber. In the same manner, if we talk about the smoke screen, in warfare, smoke screens are used, which are nothing but the colloidal dispersion of certain substances in the air. Further, it is also useful in other applications, like the blue color of the sky is due to scattering of light, and the colloidal dust particles which are seen present into the air. Similarly, the sea water looks blue in color due to scattering of light by the colloidal impurities present in the sea water. Further, tails of the comet is seen as a Tyndall effect due to scattering of light by the tiny solid particles left by the comet in its path. Blood is colloidal solution, and the stoppage of bleeding on applying the ferric chloride solution is due to coagulation of blood. There are certain biological significance of colloid also. You know that the basic unit of our life is the cell, and this cell is made up of the cytoplasm or the protoplasm, which holds different cell organelles embedded into it. The fluid part or the cytoplasm or the protoplasm, it is colloidal in nature. Now this colloidal protoplasm, it enables the movement of the cells, which could be amoeboidal movement, and this is very well known to you. In case of amoeba, the movement is possible with the help of pseudopodia, wherein the protoplasm, it enters into the pseudopodia and helps the organism to move ahead. The same is the condition with other cells which are showing movement or migration within the tissue or within the bodily fluid. So we can say that the amoeboidal movement which is shown by the cell, it is because of the colloidal protoplasm present into the cells. Also, there are different types of transportation which are seen occurring into the bodily fluid, that is blood, and this transportation of gases is because of the colloidal nature of the blood, which contains the charged molecules, and this charged molecules, it enables the adherence of O2 or CO2 and helps or enables in exchange of gases. In the same way, the synovial fluid and the cerebrospinal fluid, which is acting as a protective fluid in the joints or into the brain, there also large number of soluble particles into the fluid component enables in imparting the shock absorbing nature. Further, all the metabolic activities which are seen occurring within the body is by the action of certain enzymes and large number of cofactors. So these cofactors and enzymes, which are charged molecules, they bring about the colloidal nature into the surrounding media, and so the action becomes complete or fruitful. Now with this, we have come to an end with the topic, that is colloids, and in the next topic we shall be understanding the basics of acid and base, then pH and buffers. Thank you. Thank you so much.